Hey guys, I'm Lena.
Hey guys, I'm Kalkini.
And we're going to be talking to you about meiosis.
Yep.
So first, let's go through some basic terminology. So here, I have a chromosome, and it has two parts, or sister chromatids, that are identical, and they're attached to each other at the centromere in the center. And you can tell that this is a replicated chromosome because there are two sister chromatids. And then if you have two chromosomes that have the same genes in the same order, even though they might not be the same versions of those genes, then these are called homologous chromosomes. And we will use these for meiosis, which we'll be talking about in a little bit.
And these are all basic vocab words to understand the next application example will be presented. So here's an example of a karyotype. A karyotype just represents different chromosomes in an organism. And to start off, we're going to be talking about ploidy number. And ploidy number is basically just the amount of each different type of chromosome in an organism. So for example, this would be a diploid organism because diploid stands for 2N, whereas haploid stands for N. And you know this organism is diploid because you have two sets of each different type of chromosome. And you have 5 different types of chromosomes, and you can tell that by the different number of colors. So in this case, the haploid number would be 5, whereas the diploid number would be 10. You can tell that this organism is diploid and that these chromosomes have already been replicated because you have two sets of each chromosome. The total chromosome number in this organism is 10, and you could count that by counting each individual chromosome. And you also know it's 10 because this is a diploid organism and that there are 10, and that 2N is equal to 10. And the total number of sister chromatids, as we explained earlier, we said that each sister chromatid is half of a chromosome attached at the centromere. So in this case, there would be 20 sister chromatids.
So now we're going to go through the process of meiosis. From the 5 chromosomes that we looked at previously, we're just going to follow one through the stages of meiosis. So we're just going to use the blue homologous chromosomes. And in prophase 1, they will come together and exchange genetic information at some points called the chiasma. And this process is known as crossing over, and at this point they will form a tetrad.
And more specifically, these green circles are showing the chiasma, and this is the tetrad that's being formed.
And then in metaphase, your homologous chromosomes, or tetrads, will line up at the metaphase plate, which in the center of the cell. And then in anaphase 1, the homologous chromosomes are going to separate, and after telophase and cytokinesis, there will be 2 daughter cells, each of which is now haploid, but they are still replicated, as you can tell, with the 2 sister chromatids.
So in the beginning of meiosis 1, you have a diploid organism at 2N, but by the end of meiosis 1, you have a haploid at N. That concludes meiosis 1. So continuing on to meiosis 2, from meiosis 1, we again start off with the haploid N, and that would give us prophase, but there's only one chromosome in each cell due to the fact that there's no more duplication because you don't have interphase. So moving on from prophase 2, you have metaphase 2, and the chromosomes start to line up at the metaphase plate, right in the middle. And I always remember that because metaphase starts with an M and middle starts with an M. And then you move on to anaphase 2, telophase 2, and cytokinesis. And that's when you have your sister chromatids separating, the cytoplasm's closing off, giving you 4 daughter cells. And that also ends up with haploid N. And that's the completion of meiosis 2.